You haven't even seen the data in this presentation, Dr. Cook. You haven't even seen the data that's in it. It's gonna be Am I getting graded? That's fine. Cook. No, she hates that. She's right behind you, so don't say it. That's nice. You guys should have worn your matching ones.
We're just going to let the last of our visitors trickle in right before we get started. How was the traffic as you guys were coming up? Was there still a lot of traffic on the highways? That's no fun. Hopefully it'll be better by the time the presentation is over, and hopefully the snow stays away just in time for everyone to get home. You're ready, Sydney, we can get started.
Sure.
So welcome everybody to tonight's Superstellar Friday presentation. We are so excited to welcome you here to the Discovery Center. We have a great presentation from Sydney Rollins tonight. She's from Team Cook from Nomads. That is at the University of New Hampshire at Manchester. She'll explain a little bit more about what that is and everything that her and her team have been working on for the last
2 years.
2 years now. So it's been a good chunk of time. We're really excited to hear all of the great work and research that they've put in. So I'm going to turn it over to Sydney, and I hope you all enjoy.
Thank you. All right. So my name is Sydney Rollins, and I'm here representing Team Cook, Novel Methods of Antibiotic Discovery in Space. And I'm going to give a little bit of background so you understand the goal of our experiment, but that's who I am, and that's our logo. It's great. Okay, so in this presentation, I'm going to talk briefly about our team so you can see who we are. I'll introduce our science, give you the background on it, and then I'll tell you our methods and the kind of way we've gone about approaching the different aspects of our science. And then some preliminary findings we've gotten since we've gotten the payload back from space. And then lastly, just a brief note on our control payload as well. So first, this is our team. So in the left, you can see it's kind of an older picture of our team when it was at its biggest. So we had a lot of scientists and also people involved in the outreach side of our mission. So we try to not only accomplish our science mission, which I'll talk about in a couple slides, but reach as many people as possible and educate about our science as well. And then on the right, this is from a recent trip. We went to actually work in the labs at NASA to load our payload and then watch it. Well, you're supposed to watch it, sorry, go up to the International Space Station, but the launch got delayed. So, but we were in Florida anyway. And I promised Tom in the middle is nice. He just thought it was funny to make that face. But you'll see more of Tom. So some background on our science. Dr. Cook's lab works with bacteria. And bacteria, you may know, are microscopic organisms. They come in a lot of different shapes, and they're really small, and they do a lot of different things. One of the things they do is create antibiotics. So bacteria, just like any organism in the environment, want to compete with one another to survive. And one of the ways they do that is by creating antibiotics. You might know antibiotics as medicine you take, but they're actually, the word antibiotic just means any chemical that's against life. So bacteria, when they're competing with one another for the resources and the space in their environment, they can create chemicals and they squirt them out into the environment around them, and it kills the other bacteria around them. So in these top 3 colonies up here, you can see that there's a little clear ring around them. So in the background is what we call a lawn of bacteria, and that's where this foggy background is on top, where the letters are there. And then the 3 colonies that are in the top 3 boxes there have those clear rings around them, and that's where the chemical is, that's where the antibiotic is, that those bacteria are making. This one on the bottom here is also making the antibiotic, and it's a really strong one. So it's sending out a ton of that strong antibiotic, and that's why it has a bigger zone of inhibition around it. We identify antibiotics by the zones of inhibition that they produce. We can also extract those chemicals and then test them against that same bacteria that it originally inhibited. And that's kind of the way that bacteria, the antibiotics will get made into medications that would be safe for humans to take. We don't actually swallow giant pills of bacteria, because that wouldn't be good. So we extract the chemicals and then we treat them to make them safe for humans, and then those are antibiotics. So that sounds great, but just like bacteria can make antibiotics, they can also become resistant to them, which is where antibiotic resistance comes in. So antibiotics kill bacteria in a variety of ways, and on the left picture here you can see a lawn of bacteria that's being killed by a whole bunch of different antibiotics put on little paper discs and then put on top of them on a petri dish. So originally this bacteria is killed by all of those antibiotics. Those are big zones of inhibition, so it's really susceptible to them. But then after just a couple generations and maybe some changes in the environment, the bacteria is really not killed by those antibiotics to the same degree, and it's not killed by some of them at all. So some of those antibiotics are not effective at all anymore. And that is what we refer to now as the antibiotic resistance crisis. So antibiotic resistance has been identified as one of the biggest threats to kind of everything. And it's estimated to cause around 10 million deaths each year by 2050. So big crisis, and that is what our research aims to address. So our science mission, in the vague way that scientists have to speak, is to observe the effects of the space environment as observed on the International Space Station on soil bacteria. So our hypothesis, though, is that when we send our bacteria to space, more of them will produce antibiotics than the control payload that doesn't go to space. And we're hoping some of those will be new antibiotics that no one's ever found before, so that we can use them to combat some diseases that are becoming resistant. So the way we do that, we only had a little bit of space for our experiment, so we had to make it very compact. We used this device called an isolation chip, which was invented by Dr. Slava Epstein at Northeastern University. He was very excited to send it to space. So that is the isolation chip. It's basically a thin sheet of Teflon plastic with 384 tiny holes drilled in it, where bacteria will live. About this big. Each well, each little hole, each well gets filled with bacteria food and agar, which makes it kind of solid, and then 1 to 2 bacteria. And I'll talk about how we tried to achieve 1 to 2 bacteria in each well a little bit later. And then this is a very rudimentary demonstration of our payload setup, but the next one will be better. So basically, this is like the innermost cross section of our payload box. We put the soil where the bacteria came from. Then you put the chip, the isolation chip, with those bacteria right on top. And that way they have access to all the nutrients that was in their soil that they came from originally, and they have a better chance of surviving, because bacteria don't like to be taken out of their environment and grown in the lab. They prefer to be in their environment. So Dr. Epstein found significant increased diversity when he used the iChip as compared to standard lab methods. So that's why we wanted to use it to send to space. And then that goes down in there. So this is an animation that Tom made. Our iChip was actually split into 4 sections to make it easier to work with. All of those will go on top of, this is a little liner to make sure the dirt doesn't seep down into the bottom of our enclosure. There's our soil going down in, and then the iChips go on top of the soil. The top of the enclosure actually shows you the gyroid, which I'll talk about a little bit later. So that structure, the little shapes around the middle there, is called gyroid. And then that going in there was a data logger. So that would record temperature and humidity. And then it gets covered by a little data logger cover. Then we used our waterproof glue to seal the box up. NASA requires their levels of containment to be waterproof to protect the astronauts. We had planned to have 2 additional levels of enclosure, so there would be 3 waterproof levels, which was more than NASA required. They only required 2, but we planned to have 3. And then this is the outer nano lab, but NanoRacks is the company that kind of sponsored the SPOX project that we're part of. So this was the nano lab, the final enclosure that the box went in. So I said we planned to have 2 outer enclosures. We actually, when we went to assemble our experiment in Florida to send to space, when we had to sterilize our equipment, the boxes kind of warped a little bit. So they didn't fit in the middle one. So we just scrapped the middle one and we put it in the outer one. And you'll see that a little bit later in a video. But that was totally fine anyway. But an example of how you have to think on your feet with science. So some of you might be aware already if you're familiar with our experiment, but we didn't just choose soil from anywhere. We actually hosted a competition for middle school students in New Hampshire to choose the location of the soil that would go to space. So this was the winning team, a team from Newington Public School in Newington, New Hampshire, and they chose the Newington Town Forest. So they got to work with us for about 1.5 years now, and we're still partnering with them. And this is the Saturday before we were supposed to go to Florida. And they came out on a Saturday. All the students made it. The student teacher from last year came out too. And we spent about 3 hours sifting soil in the woods. So they were very dedicated and very excited to collect the soil that actually went to space. So that was a really cool thing we got to do with them. So here's a kind of close-up of our 4 different iChips that we had. The first iChip, we refer to them as iChip 1, 2, 3, and 4. The first one was about a 1 out of 1,000 dilution of soil in media in each of those wells. The next one was a 1 in 10,000 dilution. And we got to those, we did some testing in Florida to try to figure out how much bacteria was in our soil before we put it in the iChips. But it's really hard to count bacteria without like really expensive equipment and a lot of time that we didn't have. So we just had to make our best estimate, and we chose to kind of hedge our bets and do 1 of each dilution and hope that we would get work out to that 1 or 2 bacteria per well in the end. The other 2 iChips were inoculated differently. So we manually inoculated them with actually sewing needles that we bought from a Target in Florida because our inoculating needles didn't make the trip. So we had to improvise again. And we individually touched colonies on a Petri dish and then touched each of those wells to load the bacteria manually. So we had 2 of those. iChip 3 was loaded with pre-screened antibiotic producers. So basically bacteria that we got from that original soil, we tested them to see which ones produced antibiotics, and then we brought them with us to Florida. So we had 11 of those. And then the last one was what are called escape-safe relatives, and I'll talk more about that later. But for now, just remember that that's what's in iChip 4. So these are our 4 different iChips. I wanted to show some of our experiment setup of us actually setting up the experiment in the space station processing facility in Florida. So here is obviously a sped up version. I've got the iChip in a Petri dish full of that agar with the soil mixed in, and I'm tapping it to get all the air to come out so all the wells fill with the liquid. And then it solidifies slowly, so we have to keep it warm, and then it gets cooler as I'm working with it and as it's on that cold metal surface. And then when I pull it out, you'll see that the agar has solidified, and each of the little wells has a little plug of agar in it. And then I hand it to Dr. Cook. And she uses this sterile scraper that we've had soaking in ethanol and scrapes away the excess agar so that way we can apply our backing on it. So we use a foil backing on one side to keep the moisture in because that's not the side on the soil. And the other side has a membrane which has nanopores in it. So they're small enough that bacteria can't get through them, but nutrients can. So the bacteria can get all their nutrients from the soil underneath. So there's our
I
eye
found
chip.
this on the web.
Wow. All right. This next video is the other parts of the setup. So we loaded our bottom enclosure with soil, and then we tamped it down with a sterile conical tube there. Then we put the eye chips on top. And then Tom and Ray glued the innermost enclosure pieces together with our waterproof fancy hot glue. Then it goes into that outermost enclosure. Again, we had to skip the middle. You can actually see the discarded middle enclosure in the back corner there. It's cut because we had to abort the mission. But then this is the application of the outer enclosure. And I wanted to show you the application of the glue because I think it's satisfying. And we actually applied an extra 2 layers of the hot glue on all of the edges so that they would be extra secure. We make sure that was really waterproof. And then after that, it went into the nano lab. The video stops here because it was actually super hard to get the box into the nano lab once we got to that glue part. The rubber did not want to slide on the metal well. So we like to call it our international collaboration. We had a couple people that were there from Oxford University, and they were bigger and stronger than we were, so they pushed it together for us.
So what you're saying is you didn't want people to see you use my hand on it?
That's later. Yeah. All right, so this was our finished box. It looks unremarkable, but it has 2 years of work, so let's look at it. Thank you. All right. And then this is us in Florida, Space Station Processing Facility with our finished box. Yes, we're wearing matching sweatshirts. We went to the zoo.
Question.
Yes.
What was invented?
What? Oh, 2018? Pretty recently. You're welcome.
Is oxygen necessary?
Yes. I'll talk about that in a little bit. All right, so after the payload was assembled, we had to do a handover. So we had it stored in the fridge. Around the box right now, you can see special space tape that we were told by our mission manager is very expensive and very special. And we carried it down the hallway. This is kind of funny. We thought it was going to be, we had all thought without talking about it that it was going to be some kind of grand ceremony to hand over the box. But then Ray was like, hey, can I hold it for a second? And I was like, sure. And then the guy goes, hey, bring that over here. And then that was it. That was the handover. And then we didn't see it again until it came back from space. So there it is, being famous, taking pictures of it. And then they're going to put it into their fridge, which is the official cold stow of the space station research. And then, yeah, we didn't see it again until like 60 days later. And then, like I said, we were supposed to see the launch of it, but the launch got bumped. So we ended up watching it by Zoom, but it did go up. It went up on the SpaceX 26 commercial resupply mission. And the pod that it was in on top of the rocket is actually, that was the first time it had ever flown. The rocket itself is a Dragon, and that had gone up before, but the pod was brand new. So we were really excited that it made it there. All right, so some conditions of our experiment that we expected to see were microgravity from the ISS, some cosmic radiation. There is more cosmic radiation on the ISS than we experience on Earth. So that was a change that our bacteria were going to be experiencing. And we expected them to have internal temperatures on the ISS ranging from 65 to 80 degrees Fahrenheit. We can't actually monitor, we didn't monitor the microgravity or the radiation, but we did monitor the temperature, and I'll share that with you. Inside our experiment, the conditions were access to micronutrients from the soil that those bacteria need so that they wouldn't die. They had access to moisture and oxygen. So the whole thing being waterproof and contained allowed the air and the moisture to stay in. And that's part of the function of that gyroid structure as well, was to provide strength and structure to it, but allow for airspace. But they also had limited access to nutrients in their growth medium since each of the bacteria were only in a little tiny plug, and they had limited space. So we really hoped that would stress them out and make them compete with each other and hopefully produce some antibiotics. So that was what it was experiencing for 45 days in space. Then the payload came back. It came in this lovely box, shipped by FedEx, but it did have this cool critical space item sticker on it, which we kept. So that was fun. We cut the box open and there was another box inside, and then there was a cooler, and then we pulled it out and we swabbed all the sides of the nano lab and then of all the enclosures as we opened up the box, so that we'd be able to see if bacteria were able to travel through the walls and if our waterproof enclosure worked to keep it clean and protect the astronauts. This is where the hammer is. It was as hard to get the box out as it was to put it in, but we were more prepared. So Dr. Cook brought her hammer, the nice mallet, and yeah, and then I thought I could push it out, but I really couldn't. So Tom saves me with the 2x4. But it was, once we got it past the glue, it was all good, and we didn't have to be too aggressive with it. Yay. All right, then we really hit up the hardware store hard. We had to use a horizontal Dremel to get it out. We let Tom do that part because he had fun. So he's very carefully cutting the seam, but the Dremel was actually just melting the plastic and the glue, so he really could have done it anywhere. It didn't have to be on the seam. So he had to be very, very careful and slow with that to make sure he didn't gouge down into where the actual experiment was. And then you'll notice that he's in like a hood behind a wall there. That's called a laminar flow hood, and that's to keep things sterile. So we wanted to keep everything as sterile as possible while we open the box so it wouldn't become contaminated. So once we got the outer enclosure off, it's time to open the inner enclosure. And he had to be even more careful with this one because if he drilled too deep, he could have actually cut the eye chips and kind of ruined the experiment. No pressure. So then we used a little spatula to kind of pry the pieces apart. And then you'll see it open. We were not anticipating it to look this good. We kind of hoped, or we thought that when we opened it, we'd find the entire top gyroid just like full of soil and all the eye chips crammed into one corner, but it looked like that and it looked amazing for what I had been through. So we were pretty happy with that. And then you can see our data logger behind its little cover on the other half of that. So this is a comparison of the same photo we took before launch, before we glued it together, and then after launch when we recovered it. So you can see the data logger in both, and then the placement of the eye chips hasn't changed that much. We intended to lay them all next to each other, but there were some changes because of the warping and because of the different stock of eye chips we got. It's not a perfect process when they manufacture them. So the eye chips were actually a little bigger than we intended, than we planned for them to be. So we had to overlap them a little bit in the box. But we tried to make sure that the whales underneath had access to the soil so the bacteria wouldn't die. But that's why we were worried that they would all be squished into one corner. But only the third one and the second one kind of got squished a little bit. And there's no way of knowing if that was mallet induced or if that happened. We have no idea how to figure out, like we can't figure out at what point in the experiment that happened. So we're just kind of hoping it didn't have an impact. And if it did, then oh well. But we were really happy with how that came out. So these were the temperature data we got from our data logger. This is temperature in Fahrenheit from the moment we loaded the experiment to when we opened it in the lab again. And I'm gonna mark some little important dates here. So this was the loading date. So you can see where it kind of is low and then it spikes up. So the top spike is where we started the experiment data. This is our handover. We kind of did a little cold stove before we handed it over, and then you can see it gets warm again and then it cools way down. So this is the beginning of what we call cold stow 1. We wanted a cold stow on the front and the back end of the experiment, so all the growth that was happening for the bacteria would happen in space, and they would stop growing before and after. There's where the launch was, and it docked to the ISS, and I think it's really cool how stable the temperature is while it's literally blasting into outer space. And it's totally stable at like 40 degrees. It's like, yeah, this is fine. This is end of cold stow 1, so we don't actually know when they specifically pulled it off the cargo ship and put it into the ISS, but I'm guessing it's around this time because that's when it kind of warmed up to room temp. And then it sat at room temperature. Oh, sorry. The mean cold stow 1 temperature was about 41 degrees. And then it sat at room temperature, which was average 75 degrees Fahrenheit on the ISS, so a little warmer than you would keep your house probably, but that was kind of the average temperature with kind of a spike at the end there. Then they packed it into cold stow 2 again so that it'd be done and they would be ready to send it back to Earth. It sat in cold stow all this time for like a week before it undocked from the ISS. And then it splashed down. But again, there's really minimal temperature change while it's literally falling out of space into the ocean. So I thought that was cool. This is kind of the part of biggest variation. And this is when FedEx had it. So you can see it's in Florida, and then it comes to New England, and then it sits on my porch, and then I put it in the fridge. So it's kind of the cool journey there. And then the last little data point where it starts to rise is where we opened the payload. So you can see the temperature immediately rising when we have opened the payload. The final cold stow 2 temperature, that was a little cooler than the first cold stow, probably because of the FedEx situation. We also got humidity data, and I just want to point out a couple cool things here. I noticed these 2 dips in the humidity happen when we began and ended cold stows. So whenever you drastically or quickly change the temperature of the payload, the humidity dropped a little bit before it was able to stabilize back out to 100. So those are just some kind of cool observations. It stayed at 100 for most of the experiment, which was a really good thing because we were afraid our payload was going to dry out. It was like our biggest fear was that it would dry out and the bacteria would dry up and die, but it definitely did not. So. All right, so to get our bacteria out of the payload, we call it our recovery process. And it started with some sterile nails that we had our machine shop guy like dull down just a little bit. So they weren't totally blunted, but they weren't as sharp as the finished nails they were when they started. And Dr. Cook used a sterile hemostat to punch each of the tiny 384 wells out. And then I used a cotton swab to scoop up the plug off the nail, and then I spread it onto a Petri dish. And we did that 384 times. After I spread it out on the Petri dish, I did put the cotton swab into a broth tube, which you can see behind right here. And that was just to make sure that we got all the bacteria in case some of them didn't like to grow on solid media. They wanted to be in the broth. That way we'd make sure we didn't lose any. I like to include this picture because you can see that it's nighttime now. We were there for like 12 hours on the first day. So it was a lot of work opening the box. And this was the incubator after our first day in the lab. So like all of the Petri dishes, we filled this incubator. And when we opened it the next morning, it was stinky. These bacteria smelled worse than any bacteria I've ever smelled before. And I don't know why, but it will be in the paper. What's that? Oh, it was 26 degrees Celsius, so about room temperature. All right, so I mentioned that we swabbed all the walls of our enclosures, and these are those plates. So this was the nano lab exterior, which was way cleaner than I expected, given that I had flown from Florida and people had touched it without gloves on. But there's only about one type of mold, just those little dots are a little mold sample there. The next one was the outermost enclosure, and that also had just a little bit of mold and only on one side. And the innermost enclosure had a tiny bit of bacteria. So even though it had bacteria and we didn't want it to, it was okay because it wasn't mold, which means that it hadn't traveled from the outside to the inside. And also it hadn't traveled outside and infected the astronauts. This was the data logger. So we swabbed our data logger and then its compartment, and it was nasty. But the data logger itself was covered in mold, and then we did not find mold on the eye chips themselves. So that means that it was contained behind its little wall, which was good. All these were bacteria found on those eye chips. We swabbed the foil side and the membrane side just to kind of see what was there and make sure our levels of enclosure worked. And they did. So I want to kind of show you the process of recovery that we're currently working on and almost done with for the eye chips. And then I'll talk about the data we found from that so far. So we start with our little plug, our little agar plug, a tiny cylinder. It's like 1.5 millimeters in diameter. So it's really tiny. I said I spread that out onto a Petri dish, and I actually spread it about a third of the Petri dish and then Ray would take it out of the petri, out of the laminar flow hood where I was working and spread it out further to get it to really spread those bacteria out so we could actually work with individual bacteria. And then we made what we call maintenance lawns from each of the different morphologies of bacteria. So we tried to look at all of the plates from iChips 1 and 2 and 3 and 4 and identify all the different bacteria based on their color, the shape of their colony that they were growing in, their texture, their wetness, tried to identify all the different ones, and then culture all of those onto little maintenance lawns so that we could work with them further. We also had to observe them to see if they were contaminated or if we got two things. So once we observed those, sometimes we had to further isolate them to try again. And then we looked at them under the microscope to try to see if they were further contaminated as well. So we did that with gram staining. Gram staining just lets you learn about the cell wall of the bacteria. And if you see one color and one shape of bacteria under the microscope, then you're probably good. And if you see two colors or two different shapes, then you need to go back to the isolation step before you can move forward. So once we've got our bacteria isolated on those maintenance lawns, we organize them onto what we call library plates, which is what you saw in the first couple slides there. So that's just to help us organize them and get them onto a more compact way of further testing them. The further testing is screening for production of antibiotics. And we do that by the way that I showed you on the first couple slides, by plating a bacterium directly on top of a lawn of another bacterium, and then incubating them together and forcing them to compete for the nutrients. So some of them will grow together, and some of them will produce an antibiotic and kill the bacteria in the background. The background bacteria are not random. They were chosen by the CDC and the World Health Organization. They were selected as the 6 most antibiotic-resistant species of bacteria that we need to address, and we need to find antibiotics for them pretty quickly. So these were the 6, but given their antibiotic resistance, these are not safe to work with in the lab. They're an acronym, so they spell out ESCAPE. And then we actually work in the lab with ESCAPE-safe relatives, we call them. So these are closely enough related to the ESCAPE bacteria that if an antibiotic works against them, it will work against the ESCAPE. But if we get infected with the ESCAPE-safe relatives, we're not going to get super sick, and we'd be able to find an antibiotic that would help us. Also, we've never gotten infected, so it's fine. We also have 3 additional screening bacteria that we use to represent different pathogens besides those that were chosen by the CDC. So we have a plant pathogen, and that one actually is the pathogenic strain because it's a plant pathogen. It can't hurt us, so it doesn't matter. And then we work with Bacillus subtilis, which is related to the bacterium that causes anthrax. And we work with Mycobacterium smegmatis, which is related to the bacterium that causes tuberculosis. So those are our screening bacteria to try to fight those diseases caused by the ESCAPE bacteria. All right, so we've got our screened bacteria. We also want to freeze them so that we don't lose all of this work. If our plates were to become contaminated, or if they dried out, or if the bacteria ran out of food, then we don't want to lose our science forever. So we have to freeze the bacteria down in this sugar solution called glycerol, and we freeze them at -80 degrees Celsius, and then we can culture them back out years later. So we did that with hundreds of bacteria. All right, so iChip 3, iChips 1 and 2, remember, were both dilution inoculation, but iChip 3 and 4 were manually loaded. So they had kind of different purposes. So instead of proceeding directly from the maintenance lawn to the library plate to organize them and test them, we actually compared them back to the original bacteria that we brought to Florida with us and had tried to load them into. We're actually still working on this. So we compare the lawns to those lawns that we originally had based on their color and their texture. And then we've also compared their gram stain data to try to see which ones they are. It's very difficult because bacteria don't like to play by any kind of rules that we can understand. But we've narrowed it down quite a bit, and we're still working on that. Once those are further narrowed down, then they'll be put into their library plates and tested against the ESCAPE-safe relatives. So that data will be a little bit different. So rather than just having numbers to compare to the control payload, we'll have the actual data of, remember, these bacteria inhibited the ESCAPE-safe relatives before they went to space. And then we'll see if they inhibit the same ones after, or if they stopped, or if they inhibit more. iChip4 actually was escape-safe relatives. So we put those escape-safe relatives into the little wells and we sent them up to space. So we're gonna follow the same process to try to figure out which ones we actually intended to send and isolate those again. But then instead of testing them against the escape-safe relatives, since they are escape-safe relatives, we're going to test everything else against them. So we're gonna go back to iChips 1, 2, and 3 and retest all of those against all of our new escapes. Yes, this is taking hours, but it's cool. All right, so now I've got some real pictures of the bacteria that went to space, and our professors that walk by will call them astronauts or aliens or space bugs. So they've kind of gotten a reputation, partially for being smelly. So these are kind of some examples of iChip 1 and 2 plates, those first plates that came out of the space bacteria. You can see there's like, I wrote, first of all, there's my notations where there's which plug it was, which iChip, the date, and then notes about the quality of the iChip or the plug. So if I didn't see it, I wrote invisible, so I wouldn't know that, I would know that I wasn't expecting to see any bacteria there. And then you can see some kind of swarming bacteria here. Swarming is when bacteria kind of move over the surface of the agar as they grow, and they spread out and kind of take over. For iChip 3 and 4, we had a lot of swarming. So you can see most of these plates are actually kind of taken over by one bacterium, which was unfortunate because it makes it really hard to separate, but it's all right. We've just had to do a lot of those isolation streakings to get them separated from each other. I mentioned that we put the swab into a broth tube as well to try to keep those bacteria. We individually plated all of those as well. So we cultured them in broth and then we cultured them on plates. Based on the morphologies that we observed on these plates, they were very similar to most of the morphologies that we saw on the other plates. So we decided for our own sanity to just kind of put those aside for now and say that we had already captured it on the regular plates and we'll worry about it later, but we still have them in a fridge. These are examples of the little maintenance lawns. So we put them on smaller plates to save plastic. And you can see kind of the variety of colors that we've gotten, which was really cool because this isn't typical for soil. Like I've worked with soil for about 4 years, getting soil bacteria out of them. And normally I'll see a lot of the kind of creamish, whitish color, but not a lot of the orange. So it was really cool to see all these different colors, different opacities, different transparent bacteria. So that's just been fun as well. And then here's some examples of those gram stains under the microscope. Unfortunately, people always are like, well, what is that one? And I can't tell you what it is based on its shape. I can just tell you that it's gram positive and it's a bacillus, it's a rod shape. So most of our bacteria will look something like one of these, and then we have to figure out what exactly they are later. But we are in the process of looking at gram stains of all of our bacteria. These are examples of our library plates, so you can see them all nicely organized. The last one is we actually made some with bigger squares to allow for those swarming bacteria to have some more space without taking over the rest of the bacteria and kind of ruining the test. And then these are the most fun. So these are the actual tests against those escape-safe relatives. And if you're having trouble seeing the zones, there you go. The zones of inhibition there against that Bacillus subtilis. So that means that the antibiotics created by these bacteria might be good for anthrax. This one was created against Pseudomonas putida. So that means if you have an infection caused by Pseudomonas aeruginosa, it might help you. And then this was again Bacillus subtilis. And there's actually more that I even pointed out with the arrows, but a lot of our bacteria were producing antibiotics, so that was really cool. Okay, I have some numbers here, some preliminary data, but caveat first, this is not confirmed. We do need to do some confirmation testing, some other fill in the gaps testing, and we're not done with all this testing yet, but these are the preliminary numbers. So as of right now, from iChip 1, we have 119 isolates. We have to say isolates in quotes because we're not sure that they're isolated from other bacteria yet. We're still kind of going through those gram staining processes to try to figure that out. From iChip 2, we have 148, so that comes out to 267, some maybe different bacteria from each other. We intended to put 192, remember, because we have 296 well chips, each with one bacterium in them. We got a lot more than that, but that's all right. And then iChips 3 and 4, the 49 is just a coincidence, but those are the 49 each that we're trying to narrow down into the 11 that we intended to send in each one. So iChip 1, we are done most of the testing of iChip 1's isolates against the escape-safe relatives, and it turned out that 68% of them produced an antibiotic against at least one escape-safe relative. So that was 81 out of 119. That was crazy. We don't usually see that kind of proportion at all. I'd say we normally see like a 30%. iChip 2, we've only actually tested about 31 of them. So far a little less than half make antibiotics, but that doesn't mean that the rest won't. So I want to kind of break down the antibiotic producers from iChip 1. 44 of them were effective against more than one of the escape-safe relatives. So that means not only were they making antibiotics, but they were making antibiotics that were effective against multiple different bacteria and could help with multiple diseases. These are the escape-safe relatives that the bacteria from iChip 1 were inhibiting, broken down by kind of frequency here. So you can see Mycobacterium spagmatis and Bacillus subtilis are kind of the high hitters. Staph epidermidis, so MRSA, is caused by Staph aureus, which is the escape for the safe relative, so they might help for that. And then all the others are kind of lowish, but we've got some pretty good numbers on a couple of those. All right, so we have a lot of research left to do. Obviously, we have to finish all that stuff that I said we hadn't finished yet. And then we have further research, further steps. So from those little lawns that we have for each of our bacteria, we have to, again, confirm that they are isolated using gram stains and using those isolation streaks to try to make sure that we have one thing. And then we extract their DNA from them. And it's actually a pretty simple process now, which is amazing to me. And then we use PCR, and we do that to replicate a piece of each bacterial genome. And we kind of call it like the name tag for that bacteria. And that's because it's really different between bacteria. Yes?
How do you extract?
You basically break open the cells, and then you wash all the other stuff away, and you're left with the DNA. So we replicate that little piece of bacterial name tag DNA millions of times, and then we use a gel to make sure that we actually got that piece of DNA. And then we sequence it. So we send it off to this company, and they give us back our genetic sequence for that piece of DNA for each bacteria that we sent. Using that, combined with some data we can get from doing some biochemical tests and learning about what the bacteria can break down and eat for food, we can hopefully identify our bacteria. So that's kind of one of the goals here. We're going to focus on the bacteria that actually produce antibiotics to see if we found any new ones. But if we find the funding, we can do the rest too. I will say also, even if they don't produce antibiotics, there was a study that was done after our project got accepted by NASA that found that there was a brand new species of bacteria on the ISS, and it wasn't an alien, so that means it was something brought up by humans that then mutated so much in space that it became a new species, which is pretty crazy. So we could have some brand new bacteria that came back from space. The other kind of workflow that we need to do is extract the chemicals. So we got to figure out what our bacteria are, and then we have to figure out what chemicals they make. So we do that by actually just chopping up the agar that they grow on and putting it in these bottles, and then we freeze them at minus 80 degrees Celsius. That kind of explodes the cells and makes them release all the chemicals that they've been creating. We mix in some other chemicals, and we put it on a shaker table for 24 hours to really get all the chemicals to be suspended in the liquid. And then we separate them. They're different densities, so we separate them by that. And then we let them evaporate overnight. Then we can resuspend them in different chemicals, filter them, and then test them to make sure we actually got the antibiotic chemical out of our bacteria. So that will be a lot of work. We have not gotten to this part yet, and it actually takes, this part is probably the longest because it takes 2 weeks. We let the bacteria sit on their agar for 2 weeks so they can make enough of the antibiotic that we're able to observe anything from it. And then there's a lot of waiting steps in there, but this is down the road. All right, the last thing I want to talk about is our control payload. So The control, if you're not familiar with the control, is the exact same thing, but it doesn't go to space. So all the same steps have to be taken. It has to be set up the exact same way as much as possible. And then it just sits in this case, a classroom in Newington, instead of going to space. So we actually changed our plan a little bit. We had originally planned for there to be 2 controls. One would be in our college, and we would regulate its temperature with our little incubator to match the temperatures that it was at when it was on the ISS. And the other one would be in the school. And we were gonna kind of use the data from both, but we were gonna trust ours more. After getting to know our students, we decided that we trusted them so much and that they had been like such great help through this whole project. We wanted to give them the real control. So they actually have the real only control in their classroom right now. We set it up together with them. So you can see we've got them all gowned up with masks and gloves so they don't contaminate anything. And we had to, Dr. Cook and I loaded the iChips the day before when we brought them. And then the kids helped us load the soil in and tamp it down so it was nice and even. And then they helped us glue the box together and load it all together. And then we had the teacher push the box into the nano lab because we didn't have any strong men from University of Oxford, but he did it by himself, so it was pretty impressive. All right, and then that's all. So any questions?
When you say like 1 bacteria or 2 bacteria, does that mean like 1 or 2 species of bacteria or 1 or 2 bacteria cells?
Good question. Kind of different things depending on what I'm specifically talking about. So in the little wells of the iChip, I mean 1 bacterial cell. When I'm talking about those maintenance lawns, I mean 1 species. Does that make sense? Okay. Other questions? Yes.
Why was that so long?
Oh my God.
Because it took me 2 years to do the research, so.
That's 2 years of research right there.
Mhm.
I think it's really great that you also thought to test the exterior of the mattress as well. I was thinking, hey, I'd probably do that down stuff there. You answered a lot of my questions, but the one question that you have is you had a few slides of the antibiotic on some plates that were kind of like chart, sort of hand-drawn sort of
Mhm.
chart. Are there possibilities to kind of resuspend that into like into a plate and ship it off for any kind of drug discovery purposes? Like, for example, when you did the centrifuge and DNA extraction,
Mhm.
and you came back, this company they sent you the sequencing. Can you tell from the sequencing if there's anything there that
Yeah, definitely. So there's a couple different ways that we can do that. We work with an organization called Tiny Earth. So they kind of provide the structure of our protocols and the kind of basis of our research. And they have a system where you can send them your actual bacteria or kind of like a spun down of your bacteria, and they will do the chemical discovery for you because we don't have all of the, the lab stuff for that. And then there's also the name tag bacterial DNA is kind of a smaller chunk of it, but you can also get a full genome sequence. And that is where you can go in and actually identify the specific genes that are responsible for the production of some of those drugs. And then maybe infer that this bacterium has the ability to at least create that antibiotic. The tough thing is if there's a gene that we don't know that it makes an antibiotic because the antibiotic is brand new. So we kind of need to actually work with the bacteria, which is where the isolation chip is really important. Genetics can't tell us everything, so we need to actually culture them as well. Yes. So good question. On iChip 3, you mean? So those were producers of antibiotics. So we did our little research screening process before we brought them to Florida, before they went to space, to see if they already made antibiotics. And then we wanted to send them and see if the antibiotics would change. So we wanted to see if they still made the same ones, if they happened to make antibiotics against more things, if they stopped making them. So we wanted to see not only if they would make antibiotics, but if antibiotics that already existed would change. they make the antibiotic as they're growing anyway. So we took them. We're not sure if they were making the antibiotic when we took them. We had to separate them from that escape safe relative because we didn't want to end up sending both in the same well. But so we're not actually sure if they were making it, but we knew that they were able to. Does that make sense? Okay. Yes.
So one of the last slides where you're taking everything apart and determining after the shaking table and all those different things, you know what the chemicals are.
Mm-hmm.
So is it at that point that if you determine that this would be a handy, good antibiotic to have, you could then recreate it in bulk?
Yes. Yeah, so pharmaceutical companies will do that part. They don't want to do the part of discovery because it's research intensive, resource intensive, it's expensive. So they like to give it to students, which is where Tiny Earth comes in. And then once we figure out, hey, this bacteria makes this new antibiotic, then they'll take it. Then they're interested. Then they'll culture it in bulk and they could make the medicine out of it and get the FDA testing and all that done.
There's your funding.
Yeah.
And my second question, it doesn't say I don't have three, but two at the moment. How many proteins are there in the bacteria? They're probably different for different types, but in general, you know?
I don't. Do you know? Lots. I'm going to say lots. Yes.
Going out of the over 1,000.
Probably 10.
Like in the context of amino. Yeah, I saw amino acids go up there, and that may be pretty deep.
Yes. Oh.
Do you think that you'll try to send another box of bacteria to space?
That would be super cool, but I need time to finish this one first. Actually, we kind of half joke that our next experiment would be to put the same thing on the outside of the International Space Station so we can try to get that additional data. Yeah. Mm-hmm.
Did you all have any unexpected observations?
Like when it came back, the smell for one. We've worked with bacteria in bulk for a long time, and it was something different. It was sour. I don't understand what happened. And then definitely just the number of antibiotics that were produced was unexpected so far.
I'm sorry, follow-up question. The number of antibiotics produced was unexpected compared to the control group.
So we don't have the control yet, but compared to what I've seen in my past years of doing this research. Yeah. Yes.
So what was the main factor that you were testing for between the control period and the gravity, the radiation, most of it?
Yeah, so all of it. Basically, we had found research that said that bacteria were becoming antibiotic resistant in space. And when we hear resistance, we think, well, then that means they're competing with each other. So maybe they're also producing antibiotics. So we weren't focused on any one of the factors of space, but rather just sending bacteria to that environment to see if we could actually get them to make any antibiotics. Yes.
Do you think it would be useful to, in the future, say, one bacteria behind a test without sending them to maybe take them to the moon with a different radiation?
Sure, give us the money. Yeah, I mean, yeah, that would be awesome. I think the farther into space we could get, the like different things we could learn, but we would have to have funding for that. NASA spent about $100,000 on us doing this experiment, so hope so. Yes.
So we don't know yet what the smell will be when we go.
Correct. Yes.
But do you anticipate it being similar?
Maybe. I mean, there's no way to really know. It could just be the result of bacteria living inside of an enclosed space for 2 months, but it could be because they mutated in space. I don't know. Yes.
You think if you did the experiment again, you'd get more or less producers of antibiotics?
It's a good question. So one of the factors that I'm thinking of is the soil. So soil changes all the time, depending on the season and who happens to walk by it one day. So we actually kind of struggled to think of like how to get the soil to stay the same. We ended up just using the same soil from the same jars. But I think even in just the nature of taking new soil from the same location, because it's a different time, we might see different results. I don't know, more or less. I don't think there's really a way to predict that, especially at this stage of the experiment. But that's a good question.
So if no one has any other questions, that is totally fine.
Oh.
One more.
Yes.
Has your team thought of any ways if you were to do this again to make the process of getting the experiment ready to launch, or in terms of minimizing just the contamination off the team, putting it together, minimizing the trips to hardware stores? Have you thought of any ways to like put this thing into a seal up easier?
Yeah, so the biggest thing was that we didn't we couldn't account for the warping that would happen with our 3D printed box based on the autoclaves that we had to use. So they were different than the sterilization equipment we have in Manchester. So that's kind of where we had our tolerances all figured out with the polypropylene that we 3D printed based on our autoclaves in Manchester. But then when we brought them to Florida, there was that difference. But I think that now that we know, we could definitely account for those differences and change the tolerances of boxes. They would all fit better together. I think in terms of sterilization, maybe not use sewing needles, but they were sterile, so it was all good. Yes. I don't know. Yes. Anyone else? If you would like to stay tuned with our experiment, we've got our website. Also email me anytime. We do after school programs. Our Facebook and Instagram are the most active if you want to follow us on those. And then also we'll be sharing, we just posted a 25-ish minute video with our experiments kind of documented from finding out or applying for this experiment through the launch. And then there'll be a post one after. So we'll be sharing that as well.
Thank you so much, Sydney, for this wonderful presentation. We're looking forward to hearing all about the results.
Me too.
You could also catch Sydney at another one of our events coming up in September at our Aerospace Fest. So hopefully we may have some more information then, but we're also hoping to bring her back for another presentation once we have some more information as well. So this brings us to the end of our presentation portion of the evening. For those of you who are online, we're going to be saying goodbye to you here in just a second. But for everybody else who is here with us in person, we are actually going to have a planetarium show right after this in about, I would say in about 10 minutes or so. So at around 8:05, we'll open up the doors, bring everybody in for a night sky presentation with our very own Dave McDonald, who is right here in the front with all those wonderful questions. So we will see you down there in just a little while. Thank you all so much for joining us today, and we hope to see you again soon.
Thank you. Thank you.
We'll be done by 10:09, so just in terms of travel, so please come to the show. We open the doors in about 10 minutes, and we will be done by 10:09. 8:50, so the opening the doors will be released.
They did 2 minutes. I will say. I was just there for moral support. You know, the other just other hands. This is fun. It's been a really fun thing. Nice job, guys.
Hey, good to see you.
Show? Yeah. So much fun. How are you?
Nice job.